Okay, we need to move on to the next talk. Dr. Georg Boeck from Boehringer Ingelheim is going to talk about how to offer a nano product to pharmaceutical development.
Yeah, thank you, Bernd. How to offer nano product to pharmaceutical development. So at the end of this lecture, you should understand the process of how pharmaceutical industry evaluates in-licensing offers. You should have an idea about which teams and disciplines might be involved, know which kind of nanoproducts are of importance nowadays, and you should have learned which critical quality attributes of your nanoproduct are of interest and you might get asked for. Okay, let's go in medias res. At the beginning, a pre-evaluation will start. What is a pre-evaluation? It focuses mainly on the technical aspects, uses balanced resources of the key disciplines only, searches for knockout criteria, summarizes available data. It is a paper-based assessment and gives finally a recommendation for or against starting an in-depth evaluation. The following in-depth evaluation is very resource-intensive. It includes a due diligence visit. It is a detailed assessment of all available data. It is condensed in a comprehensive report containing contributions of all disciplines involved and addresses issues, missing data and risk associated. Yeah, finally, it ends up in a licensing proposal for decision. This is how a team structure for an in-depth evaluation could look like. So here, please be careful. The composition of the teams depends on the clinical phase of the compound and, of course, is specific to a company. In all cases, there is a kind of core evaluation team, and this nominates an in-depth evaluation team and a negotiation team. The in-depth evaluation team, or also called R&D plus M team, is responsible for the technical content, summarizing available information, associated risks, target product profiles, development plan, competitive assessment, commercial evaluation, and so on. This team splits up in the due diligence team. This team will show up at your site and is headed by a project leader and members of pharmaceutical development, analytical development, chemical development, drug metabolisms and pharmacokinetics, toxicology, and clinical development. And there is also a backup office with colleagues from patents, controlling, regulatory affairs, marketing, and operations. On the other side, the negotiation team is responsible for business-related topics for the negotiations. It's a smaller team and headed by business development, and then legal and sourcing are also part of this team. And this is the process. Also, it's just an example, and it depends also on company. At the beginning, you bring your offer to an in-licensing group, also called scouts, and they give a proposal for release of an in-depth evaluation to the core evaluation team, often based on the pre-evaluation report. The core evaluation team nominates the in-depth evaluation team. This performs the due diligence, writes the in-depth evaluation report, including the proposal, gives it back to the core team, and then the core team decides either to terminate the thing or requests additional information or releases for business negotiations. And then the negotiation team heads the licensing negotiations. And finally, hopefully, you will have an approval by the management. Now, nanoproducts. In pharmaceutical industry, nanoproducts are predominantly used for drug delivery purposes, as Olaf just presented in the lecture before. An often applied definition for nanoproduct in the drug delivery context describes particles with a particle size of less than 1 micrometers. Typically, the mean particle size is about 0.2 micrometers with a range from maybe 50 to 400 nanometers. Nanoproducts, they are also called often nanocarriers. They split up in matrix nanoparticles and in crystal nanoparticles. Matrix nanoparticles, for instance, are polymeric nanoparticles, could be amorphous compounds bound in a polymer, or the lipidic matrix nanoparticles like liposomes or nanostructured lipid carriers, solid lipid nanoparticles, and nanoemulsions. By now, the nanocrystals is the most important type of commercialized nanoproduct. And nanocrystals are made of 100% of drug, as presented by Olaf. The benefits for polysoluble compounds are to increase the bioavailability, to decrease undesired side effects after intravenous injections, to decrease large injection volumes for IV applications, and to target drugs to tissues. In the literature, you can find a blood-brain barrier as an example for a tissue. When offering a nanocrystal product to big pharma companies for in-licensing, you will get CMC questions on the compound, on the formulation of the drug product, and on the process of manufacturing the drug product. First, this is a list of key information on the compound you might ask for. Of course, you need to say anything about structure, molecular formula, molecular weight, chemical name of your compound, physical appearance, crystallinity, polymorphism, melting point. I think that's all clear. pK values, ionization behavior, apparent partition coefficient, hygroscopicity you will get asked for, purity of your compound batches used, and loss on drying, water content, such things. Particle size distribution of the batches after synthesis, solubility and intrinsic dissolution rate in aqueous media with a pH profile, very important if it is the solubility depends on the pH profile. The intrinsic pH value in saturated aqueous solution or suspension is important. Solubility in key organic solvents, surfactants, or pharmaceutical relevant excipients, and influence of mechanical stress like milling, compression on the formation of amorphous parts or modification changes. Chemical stability, of course, is also important. On the one hand side, the chemical stability under stress conditions of the solid state, here temperature, light, relative humidity as a function of time, and also the chemical stability in aqueous solution or suspension, also here a function of a temperature, light, pH, hydrogen peroxide as a function of time. And finally, of course, you will get asked for patents you have filed, the synthesis and the yields. Now the second topic, the formulation. As you have heard, nanocrystals are often diminuted with a wet pearl milling to a nanosuspension, which is then used for preclinical trials and for first in human trials. Therefore, nanocrystal products are often offered for in-licensing as a nanosuspension formulation. And the parameters I now present, they focus on the nanosuspension as an example for a formulation. If you offer a nanosuspension, you have to offer answers about the concentration of the API, how you selected the stabilizer from a qualitative and quantitative data, particle size distribution of your API in the suspension, prior and after milling, viscosity of your suspension, pH value of the suspension. Solid state of your nanoparticles, is it crystalline, amorphous? What about polymorphism? Chemical and physical stability of the nanosuspension. In vitro dissolution profile of your micro and nanosuspension. And also of the method of your dissolution testing. Do you really analyze dissolved material or undissolved small crystals passing through the filter used? Very popular question. Preclinical bioavailability data of the nanocrystal formulation compared to the microcrystal formulation, and particle size of reconstituted suspension after drying of nanosuspension. Now, this is just an example of questions you should be prepared for. And last but not least, the CMC key information on manufacturing of your nanosuspension. Here it is the equipment you used for the nanomilling, and the scale is of interest, the scalability of your process and the equipment available on the market. So if you have the choice, you should use equipment which is scalable and available on the market. Material of your milling beads, size of the milling beads, aberration of the milling beads is also of importance. The volume ratio of beads and suspension and the volume of the milling chamber. duration of the milling to get the desired particle size, kinetics of the particle size reduction as a function of milling time, change of the composition pattern of your API after milling, and change of the pH value after milling. Also, just a few examples. Finally, I would like to give you the recommendation. The list of the critical quality attributes you may get asked for is long. Create as much data as possible and reasonably, and consider the more data, the better the partner can assess your offer, and the certainty increases that a fair evaluation of your product is probably. Thank you.
Yeah, thank you very much, Georg. Do we have questions? Peter.
You mentioned the parental use of nanosuspensions. Is this really working? Because I think the solubility of these polywater-soluble compounds is not very high in serum or plasma. So nanosuspensions for parental use you mentioned in one of your slides. So my question is this really working?
It is worked because the particles are dissolved in the plasma, and therefore the volume for the dissolution is bigger, and then the dissolution can start. It's always a question of concentration. You inject a very concentrated particle, a nanosuspension, and it gets diluted, and then it gets dissolved.
And what about the reproducibility of dissolving then? Because, I mean, that will be very dependent on how you inject it and what sort of turbulences you have. So, and this is really determining the activity of the drug.
I do not see such issues because the main issue for polywater-soluble compounds in injecting them intravenously is that they precipitate. And then this becomes really dangerous for the patient. The nanocrystals, they cannot precipitate. They get diluted in the bloodstream.
But you inject them in precipitated form, so it's just the world upside down.
You inject them in a crystalline form, and it takes some time to get dissolved.
Okay.
So, yeah. It's similar to liposomes, I think. So it's similar size sometimes, and I think the mechanism is also very comparable.
What about the aging behavior of nanosuspensions in this early state? Are they stable?
You need to analyze and you need to show when you come with a licensing offer. It's of importance. And it is also important if the nanosuspension is your final product or if it's an intermediate. So if it's an intermediate, you just have to prove the whole time. But this is an important question, of course. How do you get to the nanoparticles, and how stable are the nanoparticles from the chemical perspective and from the physical perspective?
Any more questions now?